Hi everyone, it's Drew Heinmiller here. I'm a product manager at Visual Sonics, and I'm happy to welcome everybody to our live webinar today. It's called Skin Deep Ultrasound Imaging Investigations of Murine Models of Skin Disease, and our speaker today is Sarah Ryan, who's a research scientist at Biogen, who's going to be giving the talk. A few notes just really quickly. We will be recording this, and the recording will be made available a few days after the broadcast. All of your lines are muted, but we will have a question section at the end. Please use your chat window to type in questions at any point during the presentation, and then we'll be moderating that questions session at the end. The duration of the presentation today is going to be approximately 45 minutes with about 10 to 15 minutes of questions at the end. I'm happy to introduce our speaker. Again, her name is Sarah Ryan. She's a research scientist at Biogen. For the last 5 years, she's been working in preclinical imaging department looking at sort of optical and ultrasound imaging. Prior to that, she was doing extensive immunology and fibrosis work within the Discovery Research Organization, focusing on in vivo evaluation of therapeutics in multiple murine disease models. The disease model that Sarah is going to be speaking about today is a skin disease model, and this is really interesting, a very interesting topic, especially when it comes to something like a superficial organ where you really need high resolution to be able to image it. So yeah, I'm really happy to welcome Sarah, and I'm going to pass the presenter rights over to her just now. And Sarah, whenever you're ready.
Okay, I hope you can hear me. Now
Yep.
I'm just going to launch into presentation mode, slideshow. Whoops, one slide too many. Okay, hello. So first of all, I want to thank Visual Sonics and the organizers, Drew and Christine, for inviting me to come today and to share this story about our studies with all of you. So with no further ado, I want to get into the story about how we used ultrasound imaging to really help us with our analysis of a number of skin disease models we were running in mice at the time. So by way of a little bit of introduction, I'm in a group which focuses on biomarkers, and so within that group we have several different teams working on different aspects of imaging, and we think about using imaging in the context of biomarkers. How does imaging enhance our readouts of the disease or the effects of our therapeutics? We work at a preclinical level, translational levels, and clinical levels depending on the modality that we're working in, but this is always with the perspective of using imaging as a biomarker to enhance our readouts. Our group, the preclinical portion, works very closely in tight teams and collaborations with the people who are developing the diseases in various therapeutic groups, all listed here. These studies were done in collaborations with the immunology and fibrosis groups at the time. So this is very much a collaborative teamwork effort back and forth between these groups. So the last point of introduction is to say that within our team, the preclinical imaging team, we have a number of imaging modalities. So we have a multimodal team with different specialties. And as, again, in the context of using imaging as a biomarker, as we think about the disease model that we're looking at or the questions that we're asking, we can consider which of these modalities would be the most effective at giving us useful readouts by imaging. And so the story here is that it turned out that ultrasound was giving us some very useful readouts on the skin models, but not shown here. For some of these studies, we also did some optical sort of multimodal imaging on the same series of mice, trying to look at other readouts using some of these other modalities. And for this particular set of models, it actually turned out that the ultrasound was was really giving us very, very useful information. So a little bit more about the context of what we were doing. At the time that we were running these studies, we were running some other studies, again, looking at different organ areas and different models, some of these with immunology groups, some with fibrosis. And so this is really just to point out that the studies I'm going to be telling you about today are all B-mode, bright field ultrasound imaging. But at the time, we were also evaluating B-mode contrast. We were testing out a little bit of Doppler, and it turned out that those applications of ultrasound were very useful for some other models, looking at the kidney and the liver, renal and hepatic fibrosis, but not so much for the skin. So it actually turned out that we really focused on the B mode imaging. So this brought us really at the beginning in the context, it brought us to the question of whether ultrasound imaging could be used to enhance our evaluations of actually the targets that we were developing in these different models, preclinical models of skin disease. So very briefly, the technical background of what we were doing. We actually started our studies with the Vivo 770, and we were rather thrilled when we were ready to make the upgrade to the 2100 for all the reasons, all the enhancements that are part of the 2100 system. So this actually made a big difference in our analysis. Because of our applications, we purchased the 3 different transducers shown here with the stars, and for all of the studies I'm going to tell you about today, these were done with the MS700. Okay, so these slides are going to be recorded, so I don't have to go through all of this, but I did want to give a little bit of background of which types of skin diseases and models we were focusing on. So the discovery group was very interested in scleroderma, and so this is a chronic connective tissue disease, and it results in a lot of dermal fibrosis and actually damage to other organs as well, but this is actually a important clinical target. We were also interested in psoriasis as a therapeutic target, but really also for the pathways and cell types that are involved in the pathogenesis of psoriasis. So psoriasis is an autoimmune skin disease, and there's a lot of dermal inflammation characterizing this disease. So I'm going to show you data from 3 different mouse models that we were working on. The first of these is called the tight-skinned mouse, TSK1, tight-skin1 mouse. So it's a spontaneous mutation of the fibrillin gene, and this is something that was discovered years and years ago, and it's quite characterized in the literature. And these mouse quite literally have a very tight skin, so you can tell the heterozygous litter mates from the wild types because they're much harder to scruff. And it's been characterized in the literature that there's a hyperplastic thickening of a connective tissue layer below the dermis. And so there's a lot of histology and characterization of this model, but to our knowledge, very little application of ultrasound. The second model I'm going to tell you about is I'll be referring to as the tweak model. So tweak is a soluble cytokine. It's a ligand for the receptor FN14. The activation of this pathway triggers an inflammatory cascade, and it's known to result in inflammation, fibrosis, as well as tissue repair. So this is very much an experimental model. So this was using tools that we had developed in-house with a pathway that we understood well. There were numerous other studies that had been done interrogating tweak FN14 biology in numerous ways. This was unusual because this was done really focusing just on the skin. So I'll get back into some more details of this. But we found this to be quite an interesting model with a very interesting phenotype that we could see by ultrasound. The third model that we were looking at I will refer to as the Imiquimod model. This is the trade name of a clinical cream. So Imiquimod, it's also called Aldera. Imiquimod cream is a pro-inflammatory, Imiquimod cream activates the immune system by switching on toll-like receptor 7, so it's a very potent agonist of TLR7. And so what happens with the Imiquimod cream is you initiate a potent inflammatory cascade, and when this is used as a mouse model on mouse skin, it results in epidermal and dermal thickening, and it's often cited in the literature as a model of some aspects of psoriasis. So I list here the diseases that these sort of point to. As with many mouse models, they are, surrogates, and they represent some aspects of these diseases. So that's the background. These are the 3 different diseases we're going to be looking at. So a little bit more details about this because these diseases actually run in very different time courses. So first, the detail on these models is that the TSK1 is a genetic model, and so we were able to obtain these mice and perform ultrasound imaging longitudinally for up to 9 months. So it really was, there's no time sensitivity here. It was just a question of following the mice over time. The tweak model is done with biweekly injections of this soluble cytokine. So this is a subcutaneous injection of this material. And this is done over a 2-week period, and this has been defined that this was a time point that would give us some useful readouts. And the Imiquimod model is done sometimes for longer, but in our case, in our context and for the group we were working with, we were running this as a 5-day model with daily applications of the cream over the 5 days. So the pictures in the lower half of the slide are to remind me that we actually were looking at different anatomical areas of skin. And so the tweak studies were done with subcutaneous injections around this sort of red boxed area in the upper back of the mouse. And the Imiquimod cream application was done on the lower back of the mouse. TSK1 is genetic, so there's aberrations in all the tissues of the mouse, but the data that we collected, again, is from the lower back. And so skin, as we'll see in the next slide, is anatomically different depending on what location of the body you're focusing on. So a couple details about the method. As I said before, this is done with the MS700 scan head. We did 3D mode imaging, so basically we imaged sort of, 5 square millimeter region, kind of where this red boxes are indicated on this slide, and then we picked a plane of that we picked the center plane of that imaging region to do our quantitation and measurements on. The TCG settings were defined once for skin and used on the same settings throughout all of these studies. And my last point here is that for all ultrasound studies, you either work in mice with no hair or you have to deal with hair removal. And in our case, this actually ended up being a confounding factor because you need to do the hair removal in order to get optimal ultrasound imaging. But the fact of doing the hair removal sort of added some complications, which I'll get into a little bit more when I get into the data sets. Okay, so one of our major collaborations throughout the course of this project was with our partners in the pathology department. So they helped us to anatomically define here on H&E sections, just looking at the histology, just to help us understand the different layers of tissues that we might be looking at when we do ultrasound, when we perform ultrasound trying to look at the skin. So what we did is we actually took very deep tissue sections, and in both of these cases, if you start at the spinal cord, so here this is a comparison of the upper and lower back because, as I showed you before, we performed our imaging in different anatomical regions. So starting at the spinal cord, you can identify that obviously on the histology, and it's also visible with ultrasound. We can see the spine, dorsal spinous process, also quite apparent, and usually you can see some shading where the musculature is around the vertebrae. The major difference in upper and lower back is this next region. So this is the brown fat region. This is fairly unique to mice, and it's especially apparent in young mice. But on the upper back, there's this whole tissue region here, which is brown fat, which is completely absent in the lower back. So it was a little confusing when we were first trying to interpret our ultrasound images to understand which layers we were looking at. The next layer we call the adventitious, so this is sort of a fatty, loose connective tissue layer. Mice have this specialized muscle just below the skin layers, so that's one of the features that we always look for. There's then a hypodermal layer, and then the dermis and epidermis. So we were using these maps because as we went into our ultrasound images, we really were trying to interpret and understand what the ultrasound images meant in terms of which layers of skin we were looking at or what tissue layers we were looking at. Okay, so that's the background. So now to move into the very first model. So this is, again, the tight-skinned mouse. This is the genetic mouse. And this is what this mouse looks like when we performed our ultrasound images. So I hope it's readily apparent to anyone looking at this that the upper image of the TSK mouse looks quite different from the wild-type mouse. In both of these cases, the spinal cord is identified with a little white arrow, but this unusual hypoechoic area, this very dark band, is something that, excuse me, I know I was going to do that, something that we always see in TSK mice, and we do not see that in the wild-type mice. So the reasons for the histology background that I just gave you before are that we would look at these images and we could tell where the spinal cord was, and we could assume where the musculature was. And this is the lower back region for the TSK model. But we couldn't quite figure out where the epidermis, dermis, the subcutaneous layers. So we did parallel histology studies. So we looked at magnifications, equivalent magnifications, obviously, of H&E sections, and we could orient following the spine in both wild type and TSK. And so with careful consultation back and forth with our pathologists, we decided that this yellow band, this hypoechoic area, represents this sort of pinkish tissue layer that you can see in the TSK mouse, but not in the wild type mouse. And interestingly, all these different features on the histology, so the epidermis, dermis, and then this adventitial layer below, are all sort of appear almost homogeneous on the ultrasound image. But we felt that this prominent feature that we recognized with ultrasound represented these sort of bands of connective tissue layers that we could identify with the histology. So using the ultrasound imaging, first of all, we wanted to do some quantitation, and so we caliper barred the two different regions. And so this was the looking just at this aberrant band, the thickness layers. You could see significant differences in this region between the wild type and the knockout, and then also just measuring a deeper tissue layer from the outside of the epidermis down. Again, we could see significant differences. And then again, these are our interpretations of what these measurements mean translated back to the histology images. So one thing we were able to do, as I said, this is a genetic model. There's actually one study that we'd seen in the literature which was suggesting from histology that there were differences in males and females as they age. So we happened to have cohorts of both wild type and TSK mice, and we had males and females, and we actually just performed this longitudinal study. And so we could learn a few things here. So when the mice first arrive at about 5 weeks, there's not much of a pathology, but by about 12 weeks, this phenotype, so this hypodermal layer, is now quite apparent. And once it's apparent at about 12 weeks, it actually stayed relatively stable, and we took some much older mice, and it hadn't really changed all that much with considerable aging. And interestingly, although there seems to be, it's hard, but the numbers were not huge, but there doesn't seem to be much of a gender difference actually between males and females of this phenotype. But this is one of the examples of where having longitudinal ultrasound imaging on exactly the same animal provided us a lot of insight about this pathology, just to understand its changes over time. So we felt that this was a very valuable readout. So that's the extent of the first model that we were looking at and our findings from this model. Now we move to the second model, and so this is the one that I'm referring to as tweak. This is to remind you, this is where the soluble cytokine, this ligand, is injected subcutaneously twice weekly for 2 weeks, so it's a very fast model. We thought it was very interesting that as soon as we started looking at these mice, we once again observed this sort of hypoechoic dark banded region in below the skin layers, we can call them, which we weren't seeing when we injected. So it wasn't an artifact of the method by injecting any material subcutaneously, because here with an irrelevant protein we didn't see these changes. Again, in order to try to understand what we were looking at, we did a side-by-side comparison with a histology. So the tweak model, as I showed you in one of the intro slides, is done with these injections into the upper back of the mouse. And so in the upper back we have these brown fat layers, and so on the histology this is what the brown fat looks like, and with ultrasound we knew that this is the brown fat that we're seeing at this point. And these images were considerably bigger but really wouldn't fit on the slide. So the spinal cord was lined up for anatomical reference, but it's far below the crop of these particular images. So this whole story was quite interesting because we hadn't expected to see this band from what we knew before about this model. And when we looked at the ultrasound, it was quite apparent on all the tweak injected animals. And then when we looked at this super deep histology sample, we identified this band of tissue, connective tissue cells. It's hard to know just exactly what's in there, but this whole feature was actually quite apparent in this model. And this is a story of where by doing the ultrasound and by having this deep image, this whole tissue image of all of the context, we actually learned more about this model, because previously the team had been very interested in epidermal and dermal changes, and you may appreciate here, and you might see on some later slides, there's a much pinker area up here. There's actually a lot of inflammation going on up in the epidermis and dermis. So this had been characterized, and this portion of the tissue had been dissected and well analyzed and characterized, but it hadn't really been appreciated that there was this additional anatomical feature that was going on in this model. So ultrasound sort of shed light on things that may not have been appreciated before. So using this model, as I said, this is run with injections twice weekly over a 2-week period. I'm not showing all of the fluctuations here. It's rather a dizzy graph, but what we observed was that this the appearance of this band was fairly transient after each injection, and so the signal sort of bounced up and down. But looking at our data, we saw really nice differences at day 8 of this model. So this is actually the day after the 3rd injection, and so this is the point where we could see really clean differences between the isotype and the tweak injected, the cytokine injected tissues. The other feature that I'm showing you here in this slide is, as I mentioned, the fact of doing hair removal on mice where we're studying the skin caused some confounding effects. And so in this case, you can actually see that the isotype skin had changed considerably, had changed considerably by 12 days. And so we felt that this was actually a result of all of the hair removal that we were doing at this time, because the skin just appeared to be sort of more swollen and irritated because of the repeated hair removal. So again, the beauty of longitudinal ultrasound imaging on the same mice, on a single mouse over time, was able to define that the best time to run this model, the best time to look at our ultrasound images on this model was probably day 8, and if we waited till day 12, we might have some other confounding effects of the skin. So another story with this model was that we were able to demonstrate pathway specificity of this interaction. And so it's well known and well characterized that this soluble material, the soluble cytokine tweak, binds very specifically to its receptor FN14. And the group that I was working with happened to have the FN14 knockout mice available at the time, the receptor knockout mice. So we were able to run a study in which this tweak cytokine was injected both into wild-type mice shown here and into the receptor knockout. The images aren't the same scale, so I can't show you those, but the receptor knockout looked exactly like the isotype in this case. So this is another example where by identifying this measurable phenotype, which we could read out with ultrasound, and we could do our measurements pretty much in real time, very quickly we could get our information back, we were able to show very rapidly that the receptor did not have this pathology at all, so confirming that this is a specific effect from tweak acting through its receptor. So this was nice just to sort of confirm pathway specificity biology and in a really efficient real-time readout. Okay, now I'm going to move to the 3rd model that I described, and this one you can actually see right away from the ultrasound images is very different from the other 2. So this is the model in which this inflammatory cream, Imiquimod, is applied to the skin every day for up to 5 days. So there's no dark band. We're again down into the lower back, so you can see the spine here with the white arrow. These are the skin layers that we always observe, and so there's no dark band that we've seen before, but there is a considerable thickening of these outer layers when in this Imiquimod model. Again, we did a side-by-side comparison with the histology to try to interpret what's going on here. So lining up again with the spinal cord, or with the spine, we could see, we felt that these this extended region, these thickened areas corresponded to thickening in the epidermis, dermis, and even in this adventitial layer below. And so this is certainly well characterized for this model, and it's, as I said before, frequently used as a preclinical model of psoriasis. So again, we did enough measurements, so we sort of caliper measured a couple different depths and distances, and we showed that there were significant differences both with this sort of yellow bar distance of the dermis as well as this full skin distance between the treated and the control groups. And although this is a very short model, it was actually very helpful to be able to follow a single mouse again longitudinally so that we could see the development of this pathology. So on the very first day, there's a little bit of a shift. Even by day 3, you could see quite a change in the Imiquimod treated skin group. And by day 4, there really was a clear separation. So typically, the skin was often collected for this model on day 5, but we were actually able to see that, even you could get some indications even a little bit earlier on as to where these changes were happening. So the context of this model was that the group that we were working with was testing a number of compounds which were potentially inhibiting part of the processes that lead to this inflammation. So it was helpful to have some idea, first of all, of when you might deliver the compounds, when you might want to intervene, and also when you might start seeing separations to understand if your compounds were having the desired inhibitory effects. So here's an example of this type of study. So the test compound, so we had a test compound, and we did a side-by-side measurement comparing ultrasound data with histology data. And so looking at the measurements of ultrasound, so this is actually the caliper measurements and the thicknesses of these different regions. This is all based on this sort of yellow bar dermal region thickness. You could see a significant increase in the thickness of this region when the skin was treated with the Imiquimod. And you could again see a significant reduction from the disease state to a reduced state when a compound was delivered at the same time. Interestingly, with the ultrasound, you could appreciate here, even from the graph, that there's barely any significance in the difference between the baseline, sort of the healthy Vaseline-controlled skin, and the compound-treated skin that was also subjected to this model with the Imiquimod. So this was this was a nice demonstration of what ultrasound could show us. We did a comparison, so, understanding that ultrasound imaging was capturing sort of the full thickness of the live tissues, and by the time we collected these tissues and went through the fixation and all the steps of processing for histology, there's definitely a shrinkage effect. But we felt, and we also confirmed this with our pathology team, that this was very representative, and the percentages really made a lot of sense for the expected shrinkage from histology preparations. So the histology story is very similar. So again, there's a significant increase in the disease-treated group and a significant decrease when the compound is added, so it reduces. Interestingly, from the histology, the compound has not really returned as well to the baseline level. It hasn't quite reached, it's still significantly different from the control group. So it's hard to know which of these stories is more correct, but it's possible that the ultrasound story, by looking at the live tissues in real time, was able to really more sensitively show that this is almost complete restoration to baseline, or actually it's sort of a prevention of this thickening that happens in the course of this model. Okay. So the last part of our story really was just kind of a curiosity story, and it was just because we had been intrigued, as I've said all along. We didn't quite understand what these dark bands meant, these hypoechoic regions that we were seeing both in the TSK model, the first model I showed you, and in this tweak model, the second model I was going into. And so looking at this level of an H&E image, it kind of looks like it's some connective tissue and possibly some cellularity, and we consulted with our pathologists and weren't completely sure what to make of this. So we did a little bit more of a dive trying to understand how similar these two newly developed layers were, and how they were giving us sort of a similar appearance in the ultrasound. So what we did here was the H&E is on the top that I just showed you, but significantly blown up. blown up, and we're doing a comparison of control tissue, the TSK model in the middle, and the tweak model on the far right. And so H&E images are in the top panel, and the ultrasound is in the bottom panel for reference. And among other, we actually looked at a number of different immunostains and chemical stains just to try to identify what was going on here, but we felt these were the interesting ones. So Picra cereus red is a stain, a tissue stain to show extracellular matrix. And so with the Picra cereus red, you can appreciate that there's certainly a lot of a sort of pinkish banding in this region, the region that we're focusing on, in both the TSK and in the tweak models. So matrix seems to encompass a considerable amount of what's going on in this tissue region. We looked at a number of different cellular markers, and I'm just showing you one here. So IBA1 is a marker for myeloid cells. So this would show you inflammatory infiltrates. And so there's a few random myeloid cells in any control tissue, not much abundance of inflammatory cells in the TSK model, kind of randomly sprinkled throughout, and they certainly don't make up the majority of these tissue layers. In the tweak model, it has been characterized, and it was definitely reported on, that this results in an inflammatory infiltrate in the dermis. We can see that. But down here in the region that we're interested in, there's a little pocket of inflammation over here, but most of this region's not excessively made up of these inflammatory cells. So we felt that was quite interesting. This is actually just a little bit more of a blow up looking in at these regions, excuse me. We'd have to do a lot of studies and really sort of drill down on this as to whether this is phenotypically different. I mean, the TSK bands look a little bit sort of more tightly defined than what the matrix that we're seeing here in this tweak pathology. So we were rather curious about this. We would have liked to go and dig a whole lot further into understanding just what made up the different compositions of the matrix that's prominent in this region. And then shown here blowing up, again for the IBA1, the marker of inflammatory infiltrates, it's really not much of a noticeable difference between these different groups. Again, okay, so that's the end of the data set that I want to present to you. And I just really want to just stop with a few kind of bullet conclusions just to show how we felt that these ultrasound measurements were really beneficial for a lot of our models. So as I showed you in the one example, we had demonstration of efficacy. So the compound we were working in reduced the signal back down to the control level. We were able to interrogate a biological mechanism, and I mean, that study could have been done by a number of different readouts, but the beauty of the ultrasound was it was, you know, pretty much a real-time readout to understand the changes, to measure the changes that we were seeing. I went into this before, but in the second story, the tweak injections, we really didn't appreciate all of the changes that were going on in the tissues until we'd done the ultrasound studies. And until we'd seen those images and seen that extra dark band region, we really hadn't appreciated that extra tissue layer was forming. So ultrasound was actually really able to enlighten us about things that we may not have appreciated before, just because our histology might have been selective to certain regions. And of course, all the beauty of live imaging is that you are able to get longitudinal measurements on single animals, and you can get real-time data. And so we felt that these types of studies were really, this type of readout was very beneficial for helping us with our decision-making process as we develop our therapeutic targets. Okay, so last slide is my acknowledgments. So within our preclinical imaging group, I have to especially acknowledge Jessica McIntyre. She is the person who performed and did all the analysis on the ultrasound studies. So she did a beautiful job of running these studies. The next people to really acknowledge is within the pathology group. Taylor and Bob, the pathologists, were major consultants on understanding on helping us to understand what the ultrasound signals meant with the histology interpretation. So we spent a whole lot of time in tight collaborations with them. I want to acknowledge other people within the imaging group and the pathology group, and all of these studies were done in collaboration with our colleagues in the immunology group. And last of all, I also really want to acknowledge Visual Sonics, and specifically Fred Roberts, who has been a major he's been a major support for us since we've been running ultrasound studies. So he's guided us through many different methods. He's been a consultant on things. He's trained us, and so we really want to thank Fred as well. And with that, I'm actually finished.
Great, thanks so much, Sarah. That was fantastic. Yeah, really interesting. I've got a few questions here for you. I just want to remind everybody that you can just type your questions into the chat window, and I can read them out, and we'll get Sarah to answer them. A couple of questions more sort of about procedure. We got a question, someone asking, so you mentioned you're using the MS700 probe, which is the 50 megahertz center frequency probe. Someone's asking, is it possible to use the lower frequencies like the 550, the 40 megahertz transducer?
I believe that we tried those. And the signal was not as good. So yeah, I think we did some comparison studies, and so yeah, we were trying the different scan heads, and so once we'd identified these signals, we did some comparisons and determined that the 700 was ideal.
Okay, yeah, that makes sense. I mean, you with such small structures, maybe give us a sense of the again, just of the distances you're measuring here. So the thickness of those layers is around.
I'm going to go back to this slide. Yeah. Well, okay, yeah, so I think typically, I mean, in this case, this is a caliper measurement of the extended, well, actually, no, the normal sort of dermal layer, it's 0.4 millimeters. And then the dark band, the hypoechoic region that we were measuring was a little bit thicker. So again, it's sort of 0.6 millimeters, something in that. Okay. Region. Okay. Yeah. Yeah. So that I think that kind of explains it. You know, using you're measuring in the order of hundreds of microns. Certainly less than a millimeter. So using the higher frequencies to get the resolution is almost necessary at that stage. Yeah. Yeah. I think it really made a difference just to sharpen up our images and just to be able to get this kind of definition.
Yeah. Okay. Just a question about the hair removal. So we've heard this before, that the hair removal cream, typically Nair or what's the other one, Veet or something like that.
Yeah.
They do cause, they use some kind of chemical to dissolve the hair, so this can't affect the skin.
Exactly.
Did you try just using clippers to shave the hair, and then just using sort of water with gel on top or anything like that?
We did, and that was actually, it was interesting because we first did those studies with, as I said in the beginning, we had the 770. The Vivo 770, and with that we couldn't see anything when we did the clippers. With the 2100, we got decent images, but we really did get better images when we did the Nair hair removal. So we included that. We actually have a very sort of obsessively detailed protocol for hair removal. So you put on as little Nair as possible for the shortest possible time, and then you do extensive rinsing, sort of warm water, gauze pad kind of thing. So we've tried to get that to be as sort of minimally inflammatory. You want it to be as little of an irritant as possible, but it's kind of just a given for needing to get the hair out of the way in order to get the clear images. So I think what we figured out was that if we just worked within, we'd figure out when the interesting time points were for our imaging. So for example, in the first model, which is the genetic model, we could capture images once every couple of weeks or once a month and acquire a lot of data that was really useful for our studies. So you don't have to do all that frequent hair removal. And actually for the last model, it's very short, so you have to do the hair removal once and then you never do it again. And then we kind of discovered the window that we needed to stay within, because in the second model that I described, it's a 14-day study, so you really have some hair growth starting on about like day 9 or 10. And it turned out that actually, fortunately, we were able to get enough of a phenotype and enough of a measurable signal to distinguish, the treated groups from the controls. So we could just, it was an additional factor that we had to work around, but it wasn't it wasn't limiting. I mean, it didn't reduce our data acquisitions. It didn't reduce what we could understand from these studies, but it's just something to be conscious of.
Great. Yeah. Okay, thanks. I was wondering if you could just comment on and you did so generally in the presentation, but just maybe more specifically on sort of the in vivo versus histology. I actually really like, and we spoke about this before when we went through a couple of these slides, but I really like the juxtaposition of the ultrasound images with these pathology images. It sort of really outlines things nicely, but I was wondering if you could just comment on the in vivo aspect versus the histology aspect and the sort of pros and cons of each.
Sure. Well, I think our objective was, of course, to use the ultrasound, live imaging, longitudinal, and to use that as your primary readout. So that's what our complete goal was. Of course, you want to have this as something that we can follow with the therapeutic interventions, and in some cases, I'm actually not showing. There were 2 or 3 other skin models that were being run in which we didn't end up, having a useful measurable signal by ultrasound for various different reasons, like the features, the changes just weren't prominent enough to see something. So for the 3 models that I presented here, the features were apparent enough with the ultrasound that we could identify, as I showed you, all the barred regions that we're doing the measurements on for each of the 3 different models. So we had something to really focus on. But I think, the reason we wanted to understand what these changes were, it's partly to, as we're working with the discovery teams that are working within these models, just to point to the ultrasound region and say, well, I think this top layer includes sort of a fatty layer in the dermis and the epidermis. It's not that satisfying. You really want to be able to give them you want to have a complete explanation of what this signal means. So if we're using the signal as the endpoint of our studies, and this is what we're using as the readout of, in this case, the therapeutic intervention with the compound, it's very useful to have at least a theory, based on the histology comparison, to explain what this signal means, what we're actually looking at. Because really, in the end of the day, having a clear, measurable signal on ultrasound is fine, because we've already identified that there's something we can follow, and then there's something that's going to change whether or not we treat with a given compound. So that alone would be fine, but we really wanted to understand as much as we could about the mechanism and biology of what was going on. So we really had to go side by side and do some comparisons to the histology so that we could have at least a theory of an explanation, to talk about what these signals mean.
Yeah, and you're getting, precise sort of, cellular information from the histology, which you're not really getting from the ultrasound. But I found it interesting in your, I think it was actually this slide where you're showing that the effect of that thickness. So the actual measure of, or the outcome that you're looking for is different, or slightly different anyway, in the in vivo versus the histology case. And I only say that because I think, in a lot of cases, well, in most cases, histology really is the gold standard, because it does give you that cellular resolution and the ability to stain. But on the other hand, is it really what the tissue looks like when it's alive? That's the... Exactly.
And yeah, we had a lot of conversations about that as well, because the histology is a little bit of a translated interpretation of what the live tissue looked like. And so I forget what the percentage value is, but I think it's sort of established. The pathologists know what percentage of shrinkage you might have. But there's also, you've got to do some cutting to remove this tissue in some way, and then there are all the treatments and the chemicals and all of the aspects that go into preparing this tissue before it gets on the slide. So there's potential for distortion. So there's potential for there's potential for the tissue to look a little bit different than it might have looked on the living creature. So Yeah, we don't, I mean, I don't know if this would always be representative. You know, it's possible that this is, you know, this difference. So the fact that there's very little statistical difference in our treated group to the control, yet with the histology, you see much of, there's much less of a, much less significant. Well, anyway, the compound treated group is still considerably thicker.
Right.
We'd have to do this a whole lot of times to sort of confirm whether ultrasound is actually, you know, more sensitive or if it's truly more representative. But I think that just kind of intuitively, the fact that we're looking at living tissue measurements, you know, successively over time across a number of animals, it should be a more, you know, I don't know. Yeah, the measurement should represent the living state of these tissues more accurately. I guess that's about what I can say on that. So, yeah,
okay.
yeah.
Great, thanks. Yeah. So there's actually a question somewhat related. There's a attendee who's asking, how did you match up the histology with the ultrasound images? So they're saying, was it just based on spinal cord, or how did you sort of co-register those images?
Yeah, it was based on, let me go back to this one. Yeah, I mean, we did it, you know, anatomically. So you start with spinal cord and you sort of work through it. So we would look at the ultrasound images and point to sort of every single feature of the ultrasound image and then try to define what that would mean in the histology. I mean, it was definitely, you know, we did a number of different things. So we started from the spinal cord. We did do a lot of caliper measurements. So we would have our ultrasound image and we make sure that they were both scaled exactly the same, which we could do. So we knew that our microscope images were properly measuring the same millimeters as the ultrasound images. Then we applied that shrinkage factor that you would expect, you know, from the paraffin fixation and so on. So we did do a lot of sort of caliper thickness measurements. So here, you know, in order to find this thickened layer on, you know, this thickened skin layer, what's encompassed here. So we did a lot of skin measurements back and forth, and then we compared skin tissues in the histology. And this was, you know, at first it was a little confusing to some of us because, you know, looking at the histology, it looks like you've got very dense areas of the epidermis, dermis, and then this adventitial layer, which looks like it's sort of a loose fatty layer. So it looks like you've got, oh, and then this muscle layer below. So you've got sort of, you know, 4 different features of the tissue in the histology, yet when you get over to the ultrasound image, it's very hard to distinguish. Probably you can see a little bit of the epidermal layer looks a little different here, but for the most part it's hard to identify which regions you're looking at. So we did a lot of side-by-side analysis, sort of caliper measurements of ultrasound images next to similar, you know, equivalently scaled histology images. And I think that these are our, so we know what we're measuring with the ultrasound, and that's what we're reporting. And then in all cases, the pieces that I've barred with on the histology image are, this is our interpretation of what this layer means. But I can't say that we're completely, you know, we can't prove exactly what we're looking at. We're just, that's our interpretation of what this region, is, you know, in the histology.
Yeah, yeah. No, it is a nice comparison for that reason, because I think, you know, if you were just using the ultrasound image, you know, might be just difficult to distinguish some of these layers, and then you've got the histology, and then you go back to the ultrasound and say, oh yeah, okay, I see this layer here, and I see how it correlates with that. Yeah, that's nice.
Yeah, yeah.
There's another question here. Hair has repeated periods of very intensive growth.
Yep.
What specific postnatal days are better for your mouse strain?
Young mice, we didn't do super young. So I think the youngest we did, we got in the TSK mice, they're about 5 weeks old. We didn't go super young. So I think for the most part, we were working in at least 6 to 8 week old mice and then older. So it wasn't, we don't know about, you know, early development. We didn't get into that piece. So I can't really answer that. There's actually a couple of publications. So the whole hair growth cycle. Once the hair is removed, it's about a 3-week cycle till you get a rapid proliferation of the hair follicles and the next stage of growth. Although we do see some growth starting, even 9-10 days after the first hair removal. So we had to be, I think, as I was saying before, we had to be very aware of the hair removal cycles and then how to schedule our ultrasound imaging around that. And we obviously wanted to minimize the hair removal as much as possible, so we wouldn't want to have repeated cycles. But we were aware, because of what's been documented and published, that certainly 3 weeks after a major hair removal, there's a huge proliferation of new hair follicles and abundant hair growth. So that's a time, it didn't actually come up in these models, but for example, with the tweak model, if we'd wanted to look at the mice, we would have removed hair once, and then we would have avoided doing any imaging 3 weeks after that first hair removal, because we know that that would be sort of a blackout point when there would be a lot of confounding effects with the hair growth going on in the skin. So yeah, so it's actually a sort of another complexity of running all of these models, all of these skin models in the mice. It always had to be synchronized. So in any case, the control and the treated groups all had to have hair removal on the exact same cycle, because it can certainly change your readouts in the skin.
Okay, thanks. A question here about vascular imaging, and I want to expand on it a little bit.
Yeah.
There are there are different sort of functional, there are sort of different functional measurements that you can get, Doppler or using microbubble contrast for doing perfusion. Have you considered or used any of those methods?
So actually the slide, I think everybody can still see it. We found a paper, I believe it was looking at the Imiquimod model, in which they had some Doppler data. So as I said back in the beginning, we tried everything on these different models. There wasn't really a justification in the other two models. There's not really a lot of known vascular changes, maybe a little bit in the tweak model. But we started with this model because there's such an inflammatory infiltrate, and it's known to have vascular changes in the dermis and epidermis. And we'd seen this one report of Doppler. And we tried both contrast and Doppler on this model. Contrast had issues, so we were getting really nice contrast data from kidney and liver, as I said before, so contrast microbubbles. The contrast, we weren't able to put a whole lot of time into it, but what we were finding from the contrast is that there were enough bright signals already in adjacent skin areas. I'm trying to remember the set of data, but there were enough. There was enough background in the skin and in adjacent skin layers that if we were able to see any effect, if we were able to see any difference with the contrast in the disease versus control, there was no measurable difference really above the background of what we were seeing. So that was the story with the contrast microbubbles, and I think similarly with the Doppler, it just wasn't enough of, there wasn't enough of a measurable difference in the vascularity at the times that we were looking to pick up anything, measurable with Doppler in the Imiquimod versus the Vaseline groups. But we certainly thought about that. We were really excited about those. And then there's all kinds of other vascular probes and a lot of ways we could have gone with that. And I think that it wasn't, we could have taken this model out to a much, much longer model. And I think, if we had all the time in the world, we might have gone that way. It wasn't the objective of these studies. So the people who were working on these studies were less concerned about the vascularity and more concerned about the inflammatory infiltrates. So we sort of didn't have an opportunity to dig really deeply into that. But for contrast, there was sort of confounding background in the skin layers. And for Doppler, it just wasn't measurably, it didn't really separate from the control groups. Yeah, it just wasn't enough to really measure it. We were super excited about that though, but it didn't really pan out when we got down to doing the studies.
Yeah, I guess when you're getting down to that level, you're talking about really small capillaries where the flow is just so much slower that you may not have sensitivity for Doppler, and then you go with the nonlinear contrast, you're using a lower frequency probe, and it may be difficult to I'm just sort of wondering off the top of my head about photoacoustic imaging, where you could use a high frequency probe and then you're just looking at the contrast of the hemoglobin to be able to measure some differences. I know there's some groups who are sort of interested in doing that kind of thing.
Yeah, no, that would absolutely, that would be an exciting application. We don't have the equipment to do that one, but it would absolutely be exciting. Yeah, we just didn't have, there wasn't, the push from the group that we were working with to really measure the vasculature. But we, just as the imaging people on our side of things, we were very excited about it. We really wanted to try. And it just, I think that the signal, as you're saying, the vasculature is just so tiny in these areas, and the differences just weren't profound enough to really be able to measure anything.
Okay.
So, yeah.
I just want to end with maybe the last question here. I wanted to know what the clinical implications of the work are. So, for example, could you see, high frequency ultrasound being used clinically for this kind of assessment?
We had these conversations with our clinical group. And so there was a big focus. This was a while ago. These programs aren't actually ongoing right now, but there was a big focus on the disease scleroderma. So this is the human autoimmune disease. It's the skin thickening, dermal thickening disease. The word from the clinical side, sort of historically, so our in-house clinicians had generally heard that ultrasound was not a favored technique because of the usual explanation of operator variability. And I don't, well, okay, so that's actually a complicated disease as well, because it has, the skin changes are, the skin changes alter through the course of the disease in different ways. So yeah, we were hoping that group might be inspired by the studies that we were running, and they were kind of hearing from their clinical colleagues in the field, the people doing the dermatology and the rheumatologists who were studying this disease, had been a little frustrated by their ultrasound readouts, but I think that they weren't using higher frequency ultrasound at that point. I mean, this was a couple of years ago, so I think that things could have come along considerably. I think just longitudinally measuring the changes in the skin would be a great advantage for people studying that disease.
Yeah, and I think you're right. I mean, I think generally when people say clinical ultrasound, they're talking about maybe up to 10 or 15 megahertz as opposed to what you're using here, which is 50 megahertz. Yeah. So I think it's actually just a completely different realm. And I think for example, we have a clinical ultrasound device which goes up to 50 megahertz center frequency, and that's why I wanted to ask you about this. If you can measure differences in the layers of a mouse skin, I would be surprised if it wouldn't be useful in the human situation.
Yeah, I think so. And yeah, I think that the time when we started those studies, because I believe your instrument is relatively new, is that correct? So I think that the instrument,
that's right.
So the clinical instrumentation is changing considerably, and the information that I was going off of and sort of the clinical hearsay that we were basing this on was a while ago. So that landscape could certainly have changed, and it might be an option. I mean, if the higher resolution, the higher frequencies are available now, it'd be nice if somebody would check it out and see if they can measure something.
Okay, great. I guess in the meantime, this tool is sort of allowing you to develop these drugs, presumably for clinical translation as well.
Exactly. Yep.
Great. Okay, Sarah. I think we'll leave it there. I really want to thank you very much for giving a great talk and answering lots of questions. Sure. Really appreciate it. I want to remind everybody that a recording of this webinar will be available. Give us maybe a week or so to post it up, and then you'll be sent a link to be able to download this. Thank you everyone who attended, and please, if you are interested, please get in touch with us. You can go to our website, www.visualsonics.com. You can register for future webinars, check us out on social media, and we attend quite a few conferences. So if you're at any of these meetings and you want to stop by and say hello, please do so. Again, thanks to Sarah. Thanks everyone for attending, and we'll see you next time.